My name is Nimish. Uh, Today I'm going to talk about two instances where we made use of ab initio MD molecular dynamics to unravel phenomena in metal organic framework solids. Yeah, firstly, what is a metal organic framework? Metal organic framework is a material which is scaffold-like, and most of the MOFs are porous in nature. So, and they're also called porous crystalline polymers. So when MOFs or PCPs are exposed to gas pressure, some amount of gas gets into the material. So when you plot the adsorbed amount as a function of pressure at a constant temperature, you get an gas adsorption isotherm. Typically, most porous materials exhibit an adsorption isotherm as shown here in this schematic. So why do most porous materials exhibit an isotherm like this? It's because the structure of the material is insensitive to the presence of gas molecules. But in some cases, especially in MOFs, some MOFs, the structure actually is affected by the presence of gas molecules. And some among them, what they do is, when during the process of adsorption, the material as a whole swells up. So because of which a new feature comes into the adsorption isotherm, which is called a step. There's a sudden rise in the uptake amount with small change in pressure. So the advantage of having a step in the isotherm, especially in the context of pressure swing adsorption, is a small change in the pressure. You can release or adsorb a significant amount of the gas molecule, gas from or to the MOF material. So whenever we see a step in an isotherm of a MOF, it's only because of two reasons. One, because the MOF is breathing, and the other is that the MOF has large cavities where the gas molecules simply condense. So the question is, can there be an isotherm for a MOF that neither breathes nor has large pores? So the answer to this is yes. Recently, Professor Susumu Kitagawa's group at Kyoto University reported 3 isostructural MOFs which exhibit a step in their CO2 adsorption isotherms measured at 195 Kelvin. So this MOF neither possesses large cavities nor undergoes any structural change, at least no significant structural change during the process of adsorption. So the constituents of the MOF are as follows. The node is made up by zinc and the nitrate groups, while the linkers are made by halomethyl isothalate and the bipyridine groups. So authors also give the mechanism for the isotherm step, that is the emergence of new binding sites at a threshold pressure. So based on this work, we set up some simulations to understand more. This is quite a comprehensive work. We've talked about many different aspects of this material, but today I'm only going to focus on the mechanism we found through our simulations, which is totally different from what the authors proposed in their experimental work. And further, I'm only going to concentrate on the bromine PCP. That is one of the 3 isostructural MOFs which were reported. So in my study, I employ 2 methods. One is the grand canonical Monte Carlo simulations, and the other is the ab initio molecular dynamics. So there, one more thing which we have to notice, it's more convenient to talk in terms of number of carbon dioxide molecules adsorbed per unit cell rather than any other unit for the sake of convenience. So using the experimentally reported crystal structures, I calculated the adsorption isotherm using GCMC. I see that at 1 atmosphere, the saturation uptake is 9 carbon dioxides per unit cell, while the experimental observation is at the saturation of the isotherm, it's about 12 carbon dioxides per unit cell. And the onset of the isotherm step occurs at about 8 carbon dioxides per unit cell. So I'll briefly go through the simulation protocol and why I employed this protocol. First, the problem with GCMC is it assumes a rigid framework. So once I set a framework, it doesn't change during the entire course of simulation. So in order to incorporate the effect of flexibility and the effect of the gas presence on the framework, I do a combination of GCMC and ab initio MD. So what I do is I take 2 unit cells of bromine PCP, one which does not contain any guest molecules, and the other which are loaded with 11 carbon dioxide molecules. Then I perform AIMD on both these unit cells for 12 picoseconds after equilibration. So those trajectories, I call it as R0 and R11. And then what I do is I take snapshots from framework snapshots. I mean, I ignore the atoms of the gas molecules. I take the framework snapshot, and on these framework snapshots, I perform grand canonical Monte Carlo simulations to calculate the CO2 uptake amount at 195 Kelvin. Let's look at the results. So on the left-hand side, I've plotted the distribution of the saturation uptake calculated by GCMC for R0 snapshots and R11 snapshots. There we can see a clear average increase in the uptake amount. And further, to give more contrast, in one of the R11 snapshots, I see about an uptake of about 12 carbon dioxide molecules per unit cell, while the experimentally reported crystal structure shows only an uptake of 9 carbon dioxides per unit cell as calculated by GCMC. So there is about a 30% increase in one of the snapshots of R11. So the question is why? So the answer lies in the bottleneck formed by 2 nitrate groups of a pore channel. So in the crystal structure, these nitrate groups are separated by about 8 angstrom. And if we look at the crystal structures, one of the pore channels of the crystal structure, and look at where the molecules sit in the GCMC simulations, and focus on the same region of the pore, we see that there's a carbon dioxide that sits between the nitrate groups. And we have to note 2 things here. One, the carbon dioxide which sits always takes direction which is axial, and there's at most only 1 carbon dioxide sitting between the nitrate groups. And if we look at oh, sorry. Yeah, if we look at the same pore channel in one of the R11 snapshots, we see that the carbon dioxide which occupies the space between these nitrate groups are now not only radial in direction, but instead of 1, there are 2 carbon dioxide sitting there. So, and if you look at the separation between the nitrate groups, it's about 10 angstrom now. So, if I call the setup as a gate formed by NO3, in the crystal structure, there's about 7.8 angstrom. In the R11 case, it's about 10 angstrom. And if carbon dioxide needs to orient itself radially at the NO3 gate, the gate should be separated by at least 9 angstrom distance. So what I say is, here the gate is open. I say a nitrate gate is open when they're separated by more than 9 angstrom distance. And if we look at how many gates a unit cell contains, a unit cell contains 4 nitrate gates. And in the R11 case, R11 snapshots, as we can see, more number of gates are open during the course of simulation than that in R0 case. So this is actually what is responsible for the increased uptake in R11 snapshots compared to R0 snapshots. And further, the net uptake in the second step of any of the 3 PCPs reported is exactly 4 carbon dioxide molecules per unit cell, which actually tallies with the hypothesis, our hypothesis that NO3 gate opening is causing the second step in the isotherm. And one final question that remains is, we don't see any, the experiment says they don't see any large structural change of the MOF, but how can all the most NO3 gates open without actually affecting the overall crystal structure? To answer this, we'll have to look at the crystal structure closely. Okay. Yeah. So 2 pore channels, 2 neighboring adjacent pore channels are separated by buffer spaces. So whenever the flexing of these NO3 groups happen, they go into the buffer spaces. So that's why the overall volume of the crystal is not affected. So we'll quickly go through another example. Yeah. So we have a collaboration with Professor Maji's group in JNCASR. We worked on another MOF, which is aluminum based. In this MOF, there are 2 pore channels, 2 kinds of pore channels. One is the large pore channel and the other is a small pore channel. What we were trying to find out the binding energy of various guest molecules in this MOF. But whenever we try to locate good affinity sites, it always fell in the small pore channels. Actually, and the binding energies were much higher than what we estimated through the heat of adsorption. and then we did not understand why, and we just simply ran an ab initio MD on this system, and we see spontaneous closure of small pore channels. So which indicated that wherever the guest molecules go, that would be in large pores. So then what we do is we place guest molecules in various places of the large pores, large pore channels, and calculate the binding energy, binding affinities. It turns out that the binding affinities we calculated through DFT and the experimentally estimated heats of adsorption showed a pretty good match. So to conclude, in the 3 isostructural MOFs, we have cracked the mechanism of the second step in the isotherm. And in another case, we, if ab initio MD did not give us great insights, but at least it saved us a lot of time by telling us that don't work with the small pore channels, rather only look at the large pore channels. Yeah, thank you.
Thank you. Questions?
Thank you for the very good presentation. Just want to know what is the effect of basis set superposition, like BSSC energy, when you are
effect of
basis set superposition, you are calculating the adsorption energy.
Yes.
So like
we did not go through that exercise of choosing the best basis set and all. We simply followed from our own experience. We came up with a triple Z. We used a triple zeta basis set. We've continued to use it. So we have not done any extensive.
Basis set superposition energy. Have you calculated that? BSCC? No.
We have not.
Thank you.
Any other questions? You have a question? Anyone? Okay. So if not, let's thank him again and thank all the speakers in this session.